Ready for our last talk. I'm going to talk today about some research that we're doing. First, I thought I would sort of close the loop on what we do at the Bridge. So my role is to help with navigating the clinical situation. We don't do any diagnosis or any medical tests at the Bridge, as Paulette said, but I am intimately familiar with navigating the system. I know some of the doctors, and so we're very careful to keep everyone in their own lane. So if you're a Willis-Knighton person or a Shumpert person or an LSU person, we can help you navigate. You know, there's great doctors on the website, but they're not taking any patients. Or this person was there, but now they're not there, but who's their replacement? They haven't updated it. We can figure all that out for you and help you get the care that you need. We have some people that don't have primary care providers at all. We have some great people on our team for that. And the other thing that I do is research, is what I'm going to talk about today. And you'll see from our talk that there's some good drugs coming up. They're actual drug. You know, you were asking about, this comes from a squid or something. This should be great for my brain. None of those have a lot of scientific backing, but there are some disease-specific medications that are going through the FDA now. They're really close, and there's some of them that actually look good. And I've been doing this for 20 years. And I have never been able to say that. So it's pretty exciting. So what I thought I would do today is talk about, so I'm trying to get those clinical trials here for us so that we're not last in line for these kind of new innovations. So what I thought I would do today is talk a little bit about research, some of my research thrown in, but given the changes and the news cycles that are coming out about we've cured Alzheimer's disease and different things. So what I'm hoping to do is give you the tools to be able to evaluate how legitimate you think that is on your own. Because the situation is very complicated, and I don't think any one person is going to cure Alzheimer's disease. So that's the framework upon which I put this talk together. So some basics. I'm going to start with what is a biomarker. This is sort of the crux of all of the research that we're doing. We're looking for a biomarker, which is defined as a measurable substance in an organism whose presence is indicative of some phenomenon, such as a disease, and that's what we're interested in, a disease. So what a biomarker is it's a disease, it's a target that we can go after. So, for example, if you have strep throat, you go to the doctor, they do one of those horrible swab things, they put it on a dish, a specific bacteria grows, and we know that you have strep throat. You get an antibiotic, you take the antibiotic, that bacteria goes away. So being able to measure that bacteria tells us about the disease process and the effectiveness of the treatment. So that's a pretty simple one. What we're looking for is something for Alzheimer's disease. And it turns out that Alzheimer's disease has several important contributing factors, and those factors are differently weighted in each individual person's disease. So I'm going to talk a little bit about three of the big ones, talk about what kind of biomarkers we have, and then how that relates to what might be coming up in the press. One other concept that I'll refer back to is the idea of sensitivity and specificity. So we have a marker of Alzheimer's disease. We want to know sensitivity, or how well does the test identify people who have the disease. So if I have a treatment, I want to identify everybody who has the disease. I don't want to leave anybody out. If my test is only 80% sensitive, then out of 100 people, 20 people will be left out of my treatment trial. So that is not a very sensitive test. We like sensitivity up in the 90s, where we don't leave anybody with the disease behind. Specificity is how well does the test identify people who do not have the disease. The other problem is if I have 100 people with Alzheimer's disease and my test says that I have 120 people, then 20 people that are in the trial don't actually have the disease and will not benefit from the treatment, okay? So these are the concepts that all of our scientists are trying to find for pretty much every disease, but neurodegenerative diseases in particular are difficult. And I'm going to explain some of why that is. So I'm going to go over some background. So 3 of the contributing factors to Alzheimer's disease that we're all scrambling to find biomarkers for. Stacy mentioned beta amyloid, or the amyloid hypothesis. So beta amyloid is a protein that folds up, that is misfolded and clumps up in Alzheimer's disease specifically, that forms plaques. So probably most people that are interested, who know anything about Alzheimer's disease, know that we get plaques and tangles in the brain. So one of the areas where biomarker discovery has been pretty good is finding beta amyloid. The other protein that we're interested in is tau. Those are the tangles. That biomarker discovery for tau is a little bit further behind, but still getting better. I'm not going to go into the details there. And then there's a vascular contribution to Alzheimer's disease as well. This research is a little bit newer. This is actually where my research is coming from. And as Stacy said, I think all the stuff that they tell you about heart disease also super important for your brain and probably contributes to developing dementia. So the way that I think about it is your brain is very fragile, and it's in a very delicate environment inside your blood-brain barrier, and the older we get, the more fragile it is. And these having atherosclerosis can deprive your brain of oxygen. Having beta amyloid or tau also stresses out your cells more, and so it exacerbates that increasing fragility and results in cell death more easily than a brain without beta amyloid, tau, or atherosclerosis, for example. If anyone has any questions, please feel free to ask me. And we can have a question and answer session at the end too. Okay, so this is a timeline that's come out recently, 2019. And what you can see is that over time, the first thing that we think that this cardiovascular disease in the brain vasculature is early in time of disease progression. Then we see beta amyloid and tau, which results in cognitive impairment. So it's a sort of more complicated than it needs to be, but it's a timeline showing first we think cardiovascular issues, which accelerate beta amyloid clumping and tau accumulation, which then impacts cognitive function. So you can see from there, if you want to fight Alzheimer's disease, like Stacy was saying, increase your steps. I like the inefficiency. I had never thought about it like that. Diabetes, try not to drink too much soda, those kind of things. Okay, so one of the best or most recognized signs of Alzheimer's disease comes from brain imaging. So up here, we have a normal brain. You can see here's the eye, so this is a side view like this. And you can see these are the ventricles here. This is where your cerebrospinal fluid is. And you can see that the brain looks pretty full. There's not a lot of empty space in here. And here's someone with dementia. So they've got enlarged ventricles and some empty space. And that's from brain atrophy. So brain atrophy is a marker of cognitive decline in Alzheimer's disease. So I'm going to go through some of the markers now that we've reviewed what the biology is in terms of tau, beta amyloid, and vasculature, and now I'm going to tell you what we can measure pretty well and how well they work. For MRI, we know that these changes are pretty early and they're pretty easy to see. You can easily get an MRI from your doctor and have that read by a radiologist. There are also brain imaging techniques that can measure beta amyloid and tau. These are called PET, or positron emission tomography. They involve a radio tracer that labels beta amyloid or tau, and then we can measure that radioactivity to tell if where and how much beta amyloid or tau is accumulating in the brain. So for beta amyloid PET, it has a low false negative rate. That means if they tell you your scan is negative, it's really negative. But it has a high false positive rate. What that means, what I think that means, is that there are a lot of people walking around with a fair amount of beta amyloid in their brain who are fine. They don't have dementia. So we don't fully understand that. So what you're gonna see from this is that what we know, they're all probabilities. So if you have beta amyloid positive scan, your probability of getting dementia is higher, but it's not 100%. None of these tests are 100%. It's actually maddening as a scientist. If you want to study Alzheimer's disease, you really want to be able to say, this person has Alzheimer's disease, and then study them. But what we're doing is like we're betting on horses. We're playing the ponies, because all of these things are probabilities. So looking longitudinally at beta amyloid, they were able to classify who had Alzheimer's disease at about 89%. That's much better than flipping a coin, right? But for an individual person, that extra 11% is still a pretty big window that you might not have Alzheimer's disease or end up with Alzheimer's disease. Does that make sense? And with tau, it's almost the opposite. So there's the tau, PET scanning for tau is a little bit newer, but it has a sensitivity, it has a specificity of 80%, but a sensitivity of 38%. So it's missing a lot of people. So if you remember what we were talking about back with sensitivity and specificity, it has a sensitivity of 38%, specificity of 95%. So if you have a positive tau scan, you probably are having memory problems, but it's missing a lot of people. And here's just a picture of a tau scan to try to make it a little less dry. Here's folks with dementia. You can see these bright colors means they have plenty of tau. Here are folks that have positive beta amyloid and mild cognitive impairment. They don't have much, they do have some tau, but not a lot. But these folks are almost certainly on their way to Alzheimer's disease. And then here's in non-Alzheimer's disease disorders, there's still a little bit as well. Okay? So ideally, these scans are very expensive. Your insurance doesn't like to pay for them if they don't have to. There's some arguing going on right now about whether Medicare is going to cover beta amyloid scanning. They're trying not to, but we're actually a site for a study where they're trying to figure out if that's financially and clinically feasible, important thing to do. And the field is moving toward blood tests. So a lot of work is being done on the blood tests, but they also do not have perfect sensitivity or specificity. And in fact, it's a little bit worse than the PET scans. But it costs a fraction of what a PET scan costs. Okay, so those are our information for beta amyloid and tau. So now I'm going to talk about that vascular contribution, and that's the study that we're doing at LSU, and we're always looking for more people who are interested in doing it. We're about to start a new trial. We do an in-depth memory screening, an MRI. We have a radiologist on our team who will read the MRI, and we can give the information to your doctor. I will reiterate that we do not give our results directly to the participant because I am not an MD. We are not allowed to do that. But we put it all together, give it to your care provider, your primary care provider, your neurologist, and they can pass it on to you. And they're getting used to that. They're getting used to us now. They don't lose our facts as much as they did in the beginning. Okay, so the molecule that we're looking at is hydrogen sulfide. It is, in fact, the thing that makes eggs smell bad, but it is also a chemical that is used as a messenger inside of our bodies. And one of the key things that it does is, at normal levels, it's a vasodilator. So that's good. Most of the literature says that hydrogen sulfide is good for you. You know, as we get older, our vessels get stiffer. We get sleep apnea or snoring, so we're fighting against increased risk of oxygen deprivation to the brain, which, as you can imagine, is bad for those fragile brain cells that we're trying to protect. So we collected from blood folks with and without memory issues, and what we found, this is a scatter plot. Here, this is their cognitive score. So higher number is worse, and here is sulfide, and you can see there's this linear relationship. So the folks with higher sulfide had worse memory scores, and the colors, the folks who are a dot that's filled in, they had a memory score that was pretty good, and we had a certain cutoff for dementia and not dementia, and so these open circles are the folks who scored above that cutoff and had possible dementia, and you can see that they almost all have much higher hydrogen sulfide. Here's a measure of an MRI measure of cerebral microvascular disease. So if you have a higher cerebral microvascular disease, you also have higher hydrogen sulfide. This is a picture of an MRI. You can see these bright white spots. Those are the lesions that we're looking at that are indicative of microvascular disease, so clogged tiny vessels in your brain, which is bad, obviously. The other thing that we found was that this brain atrophy that we talked about was also linearly associated with increased acid labile, hydrogen sulfide. It comes in several different forms, not super important for this talk, but so bigger ventricles and also more atrophy in the cortex, both were associated with higher hydrogen sulfide. We found a cutoff point. So here's our specificity was 93%, and our sensitivity was about 0.8. So that's pretty good. It's better than some of the beta amyloids tests, blood tests especially. And then here's just a picture of, we used that cutoff point to look at cortical thickness. So this is a brain from the side view, so the eyeball would be here. It's the left and right hemisphere. And where there's more red is where there's a bigger difference in cortical thickness for the high and low sulfide group. So it's a little bit different way of looking at it. But the point is, here's the temporal lobe where the hippocampus is, and it is bright red. So it is shrunken in people who have high hydrogen sulfide. And we just published this a couple months ago. The paper came out. Okay, so I talked about that. So what is the point of me showing you all these tests? The future is going to be personalized or precision medicine. So each one of us has different numbers for all of these tests, and it's going to become more and more important as these drugs are coming out that we know our numbers. So the really exciting thing that's happening in Alzheimer's is a lot of drugs are coming out that dissolve that beta amyloid clump. They've been in the news, the FDA is fighting over them. The first one that came out had a lot of pretty bad side effects, and half the FDA panel resigned. I mean, it was a brawl about what to do about that drug, because of course we're all desperate to have a drug that actually does something. On the other hand, Medicare doesn't want to pay for a drug that we would all end up taking that doesn't work. And so that's where both sides had a valid point, and they sort of split down the middle. And but what's happened since then is another drug from Lilly has come out that is an infusion that is much lower side effects. They went for rapid approval by the FDA, but were denied because what happened was they were giving the drug to folks and their beta amyloid went away. So they had set up the protocol to give 5 doses or something. And after 3 doses, it was better, so they stopped. The FDA said, well, we don't have that longitudinal data, we're not gonna give you rapid approval, but still pretty exciting. So they went back, I mean, they're still fighting the paperwork and the, but that's right on the horizon where we can get a drug that, and then the cognitive improvement looked good. I mean, we're right there. The problem is, if you don't have elevated beta amyloid, if you're one of those 10% or 20% that don't have elevated amyloid, this drug isn't going to work for you or your loved ones. So it's really important going in, watching the news, talking with your doctor, that you understand that there's drugs right behind it coming for tau. They're not as well, they're still either doing animal studies and they're not to the same phase of development, but those are coming too. And so if you're a tau person, you need to wait for the tau drug. So that's sort of the point of why I wanted to go through all of this, so that you have the background and the tools to look at these things coming out and talk to your doctor in a way that could be effective for you personally to figure out what you should do. Should you put your mother on this drug given what the side effects are? And the other thing is, you can call us and we can talk to you about it too. All right, I think that's it.
Dr. Disbrow, how often is the infusion on, like, with beta amyloid?
You know, that's a good question. I don't know, I haven't looked at their study protocol for that one. Sometimes it's hard to get them.
And you have to have a very distinct set of clinical criteria to qualify.
Right, you have to
correct?
qualify. You
Yeah.
have to have elevated beta amyloid. Which brings me, okay, so what do you do? Like, how do you figure out, can I do this? And when we are not a site, okay, I actually fought with the neurologist. There were 2 choices. There was the Biogen drug, and there was the Lilly drug. And I happen to be friends with, I've been working for many years with someone who's risen in the ranks in Lilly, and I talked to her about their drug, and I said, oh my gosh, I want that one. Well, the neurologist that I work with had been talking, has a similar situation with Biogen, and he's the one that's doing the study, and he signed up for that one, and that one's not gone forward yet. But I'm gonna keep working on him, because it's pretty clear that Biogen's gonna be dead in the water, and we're gonna, so please stay tuned. I'm
That's
trying to
the
actually get us that drug.
That's the Aduhelm one. The Biogen is the Aduhelm
Yes,
that you've heard about.
yes, that's the one that,
Aduhelm,
Oh, yeah.
that's the one
that's the one where the
that's been in
FDA
news. Yeah.
exploded, and
Right.
yes, over it. And the side effects were pretty significant. There were a lot of neuroinflammation, people were getting hospitalized from the drug. So, you know, my rule of thumb is, would I give this drug to my mom? Maybe not on that one. So then the question is, how do I figure out, should I be in one of these trials or not? And in your packet, we've made this bookmark in the bags that you're gonna get. We've made this bookmark that has these tests on it. So here's beta amyloid and tau. These are not standardized tests that your insurance will pay for, but they are commercially available. So Megan Harris at Willis-Knighton actually is the one that told me about this. I think it's about $1,200 to get the blood test, but it is available now. It's not something where you can just go to the lab down the street and get it, but you can get it, especially if you know to ask for it. ApoE status, we have a geneticist with us. This is the main gene that we know about that is related to Alzheimer's risk. It's a gene for lipid or cholesterol transport in the brain. You have 2 copies from your 2 parents, and the different forms have been labeled 1, 2, 3, and 4. 1 and 2 are pretty rare and not particularly bad for you. I think 2 is actually lowers your risk. The majority of the population has two number 3 forms, and that's standard, and that's the baseline for Alzheimer's risk. If you have one copy of the 4 allele, that increases your risk somewhat, and if you have 2 copies of it, that increases your risk more. So you can, I think 23andMe will do this. Is there another way they can do that here?
So remember, for the all that do 23andMe, it's recreational. You can get it done, but it has to be then confirmed with a clinical diagnostic test. No, that is, and it's hard. I don't wanna go, it's not my gig. So I will tell you that ApoE4 though is very hard. Chris Helm, you know, Thor, he just had a big public service announcement. He was diagnosed, he had ApoE4 and he went through this whole thing, what he's doing to prevent Alzheimer's. If y'all see, I'm sure y'all seen on that. So that's been a big question for a lot of my patients that come in. But yes, it is a commercially available test. You can order parts of it through 23andMe, but that's not the whole picture. So just be careful if you get your results back and you're running around saying, oh my gosh, I'm ApoE4, I'm gonna have Alzheimer's, because it does need to be confirmed.
Right, and to put it in perspective, about, I think 25% of the general population has an copy of the ApoE4, but in Alzheimer's disease, it's about 65%. So again, we're playing the ponies. It does increase your risk, but it's not like other diseases where if you have this one genetic change, you will 100% get the disease. It just raises your risk. So one of the things that I've heard doctors say is that you would more aggressively control your cholesterol, maybe with a drug where normally you wouldn't, so to keep those levels down more and try to get your risk back down into the range of people who don't have an ApoE4 gene. So these are all lipid panel, blood pressure. Homocysteine is an inflammation marker. We've talked about thyroid, vitamins. This stop bang is an interesting one. I was just down at the Capitol last week testing all the state legislators. It's just a series of questions. Do you snore really loud? If you snore really loud, you might talk to your doctor about getting a sleep study, is the upshot of it, especially if you're a man and you have a neck that's over 17 inches. So, and you're over 55 or 50, something like that. So if you're a little bit older, you have a big neck, or if your spouse is pissed off because you wake them up all the time with snoring, you might talk to your primary care provider about getting a sleep study. And Willis-Knighton and LSU both have centers where they'll do that. And insurance will usually pay for some form of that test. And what they do is they measure your oxygen with a little clip on your finger to see if your saturation goes down while you're snoring, to see if you're depriving your brain and your heart of oxygen, because obstructive sleep apnea increases your risk of stroke, heart disease, and eventually dementia as well. So this is, so if you want to talk to your doctor about any of these, the bookmark is in your bag, and that is my message today. Are there any questions? All right.
Thank you, Dr. Disbrow. Every time I go into her lab and she talks to me and I step in there, I feel like my IQ goes up, and then as soon as I step out, it goes back down. But thank you for that information. So thank you all for coming out. We're going to give you a bag. We're going to stay back for a little bit. We ended early, so that's fine. You can enjoy this beautiful day. And please call us if you need us. You'll have all of our information on that. If you have filled out a half sheet, we will pick those up from you, and we will stay and answer any questions you may have. And again, thank you so much for coming out.